In these slides, I mentioned some oncoantigens. They are good targets for immunotherapy. So I thought, why not pick one of them and do some research about it. So I chose crypto1. It's an oncoantigen that has been revealed to be a good immunotherapeutic that can respond to immunotherapy. So I found this paper entitled Crypto1 as a potential target of cancer stem cells for immunotherapy. So I'll start why attack new targets for immunotherapy. Mainly, there is many reasons. Three of them is that no immunotherapeutic target has been found that retains validity in the face of tumor diversity. So we saw that cancer can be very heterogeneous. So not every patient have the same antigens, have the same markers. So it depends on every patient by itself and every histotype by itself. So it's good to just do some research about the new targets for immunotherapy. Secondly, only around 5 to 30% of all cancer patients who receive immune checkpoint inhibitors as immunotherapies effectively respond to it. So this is a relatively low number for immunotherapies. So these ICIs, which we talked about, like CTLA4 and PD1, PD-L1, not every patient will respond in the same way and with the same efficacy. So it's always good to target new antigens. And strategies aiming at boosting the immune system that attack cancer were not successful in every patient. So as we said, there are other types of immunotherapies which can just boost the immune system to help it attack cancer cells. So this therapy also was not very efficient in providing a cure for cancer. So in this article, they mentioned the TGF-beta, which are transforming growth factor beta. These are it's a cytokine family that possess a broad biological activity. So cancer growth is regulated by various cytokines expressed in the tumor microenvironment. This includes the TGF family. So why they thought to target this precise family? Because it has a broad biological activity. It can be involved from the induction of helper T cells, which are important in immunotherapy. And on the other hand, they can suppress the anti-tumor response by inhibiting the infiltration of T cells into tumors. And they can promote stemness of cancer stem cells in malignancies. So they can promote malignancy and can promote the differentiation of cancer cells and the growing of the tumor. So these are good targets for immunotherapy. This family of cytokines is relatively large. So they found that crypto1 is included in this family. Crypto1 is a unique co-receptor required for nodal activity. Nodal is a member of the GF family, which plays an important role in cancer stem cells, enhancing tumor growth and metastasis. So nodal is actually a co-receptor. I saw a video about how this nodal and crypto1 will interact to cause their effects so it's a pathway. It's difficult to say how it works. So I did not include some pictures and graphs. I had some difficulty with time, I told you. Okay, just one. Okay, so crypto1 is overexpressed by various types of tumor cells. They found that crypto1 is expressed in different types of cancer, from breast, colorectal, gastric, ovarian, lung, pancreatic, basal cell carcinomas, bladder carcinomas. So it's a huge amount of cancers that this antigen is expressed in. So it's a potential target for immunotherapy. It is mainly involved in breast cancer tumorigenesis and found to have an essential role in angiogenesis, which is the development of blood vessels for the tumor cells. So it is a good target, I think. They did a number of studies on this oncoantigen, and there are different results. Some of these findings is that crypto-1 vaccination was effective in inducing protective immune responses against metastasis produced by sphere-forming CSCs of melanoma and mammary carcinoma cells. So in this article, they included a lot of studies with different type of cancers. I mentioned some of them, and we can find in this article that they mentioned some study talking about the suppression of crypto-1 expression inhibits cell stemness and the MT-related gene, which is this gene can cause metastasis. So reducing self-renewal capacity, tumorigenesis, and metastasis esophageal squamous cell carcinomas. And one more study I mentioned is that monoclonal antibodies against this crypto-1 interrupted the signaling of this pathway and resulted in gross suppression of both colon and testicular cancer xenografts. So it is it can inhibit the growth of cancer by inhibiting the signaling pathway that this co-receptor can be involved in. And they mentioned a lot of more studies about crypto-1 and the inhibition of TGF beta cytokines, and they found relatively good results and efficiencies. So that's basically it about this crypto-1 immunotherapy. And I included in the last slide, if you remember, I told you that the first study you told me to read about collagen 12 alpha 1, I told you it was a bit hard for me because it included a lot of information from bioinformatics if you read the article. So I had some difficulties reading it, but I got the main idea of this article.
Okay.
So they said that they can find some biomarkers that can be related to the prognosis of cancer patients. If you have this specific gene, or if the expression of this gene is involved in your type of cancer, they can say if your prognosis is good, if your response to treatment is good and long term. So they found that a lot of genes can be involved in prognostic biomarkers, but they put an importance on this one, which is collagen 12 alpha 1. It potentially serves a critical function in tumor immunity. And they mentioned some results from different studies also in this article. They said that expression of collagen 12 alpha 1 predicted poor response to anti-PD-1, PD-L1 therapy.
Okay.
So it's good that they found this specific result. So they can screen for this biomarker before involving the patient in this therapy, because it would be useless if the patient will respond poorly to this immunotherapy. Another study said that higher expression of collagen was correlated with enhanced chemotherapeutic resistance. Also, it can resist to chemotherapy, and increased expression of collagen 12 alpha 1 indicated unfavorable prognosis in breast cancer, specifically breast cancer.
Okay.
So briefly, I can say that collagen 12 alpha 1 represents a promising biomarker for prognosis and immunotherapy response prediction of breast cancer patients.